And our next speaker, as Mario comes up to the podium, Mario is professor of internal medicine at Yale University School of Medicine and Yale New Haven Smilow Cancer Center. He's the clinical translational research leader of the melanoma program and co-director of the Yale Spore and Skin Cancer. Mario began his education in Houston at Rice and Baylor, did his medical training, medical oncology training at Mount Sinai, and then he spent 12 years at CTEP. Before immuno-oncology was a hot topic, it was probably more controversial than it was hot. And now he's part of the Yale program making significant advances. This talk came out of our conversations in regards to planning the translational medicine retreat. And we thought that we had to have a combination therapy talk, but it just wouldn't fit into the time frame or the theme of tonight's retreat. So here we have the pleasure of hearing Mario teach us about this during the day. Thank you, Mario.
So thanks, Lee, and thanks for the invitation. My job is really to set up the symposium for tonight and the problems that we're dealing with in trying to get through this immense complexity. So, by now everybody must know this, I would think, because I've given this talk 6,000 times, but I need to go through it again because everybody should be up to speed with the biology. So, in the context of tumor development, proteins are released. They're probably taken up by antigen-presenting cells. And in that process, those antigen-presenting cells need to be activated in some way. And they present the peptides, which can either be presented to CD4 or cross-presented to CD8 T cells. You generate a T cell response, and theoretically that T cell response can which probably occurs in the lymph node, can then travel back and either control the tumor, or the tumor can grow in spite of that T cell response. In fact, there's some data to suggest that inflammation helps tumor progression. But the key points I want to make here is that when these peptides, which are presented on MHC molecules, are presented to T cells, in order for the T cells to become activated, they need co-stimulation. That's usually a ligand-receptor interaction between the antigen-presenting cell and the T cell. But just like everything in biology, when you have an activation signal, you also have a negative regulatory signal. And when, at least in the simplest model, when this T cell becomes activated and starts making cytokines, CTLA-4 is brought to the surface to the immunologic synapse, binds to the same ligand, B7, that bound to the co-stimulatory molecule, binds it with higher affinity, soaks it up so that it's not available anymore, probably transmits a signal to the T cell and dampens T cell activation. So that's at the level of T cell activation. But when the T cells get to the tumor microenvironment and they're activated, they upregulate PD-1 when they see their antigen. And they also make cytokines. Those cytokines upregulate the ligand for PD-1, either on neighboring immune cells or tumor cells. And that PD-1 interaction basically turns off the T cells, dampens its function. So a lot of what's been going on in immunobiology in the past 10 years has really been either providing co-stimulation or, more importantly, blocking the negative co-regulatory molecules like CTLA-4 and PD-1. So when you look at tumors, they come in all types, as you would expect. There are tumors that have no lymphocytes in them at all, like this one or this one. And there are tumors that do have lymphocytes, and in some of those tumors, PD-L1, the ligand for PD-1, is expressed close by, and in some of them, there's no PD-L1 expression close by. But then, as you can see, there's different flavors of these, and depending on the tumor type, this is work that was done in part by the people at Hopkins, in part by Kurt Schalper in our group, who'll give a talk tonight. What you can see is that different tumors have different subsets, different percentages of these types of tumors. We would guess that most of the patients who respond to anti-PD-1 are in this group, but we don't actually, and you'll see it's a little bit more complicated than that. But you can imagine that you might need to address each of these groups differently with different types of immune therapy combinations. So it turns out that when you, for example, in melanoma, you go take a deeper look and you take out these T cells and you just sort them by expression of PD-1 or not expressing PD-1, and then you grow these cells in vitro for 15 days and then you test them against autologous tumor. it's only the subset that express PD-1 or some of the other regulatory molecules like LAG3 or 4-1BB that actually recognize tumor. The PD-1 negative population does not. And that means is that not all the T cells in the tumor microenvironment are tumor antigen specific. They're probably just bystander cells. The ones that are probably PD-1 positive, they don't function very well when you take them out of the tumor fresh. But if you culture them in vitro for 15 days, they do work perfectly well and can kill and secrete cytokines when they see their tumor. And this is the kind of data, this data was only generated a couple of years ago, but this is the kind of data that supports looking at PD-1 or PD-L1 antagonists in the clinic. And just to cut to the chase, this is just a list of all the tumors. I haven't updated this in a while, but it's clear that anti-PD-1 and anti-PD-L1 has activity in a large number of tumors. It's amazing, and it's not 100% in all these tumors. In some cases, it's only 15 to 20%. In some of these tumors, you get dramatic responses that are very durable, and some not as much. But still, there's activity across a broad range of malignancies, and in fact, these days we talk more about the tumors that have no activity or very little activity versus those that in the old days when we would talk about the ones where the few tumors where we would see activity of immune therapy. And this has led to this immense development of not only PD-1 and PD-L1 antagonists, but to many immune therapies in the clinic. There are already now four PD-1, PD-L1 antagonists approved in the U.S., nivolumab, pembrolizumab, atezolizumab, and most recently, avelumab. Who responds and who doesn't respond to these agents? And I'm not going to go through all that because that's the purpose of tonight's symposium. But suffice to say that it would be obvious that we would look for the expression of the ligand for anti-PD-1 or anti-PD-L1 in the tumor microenvironment. And if you do that, and this is in the, I apologize for the quality of the slide, I stole this. But if you look at the, this is now in the Keynote-06 trial, which was pembrolizumab versus ipilimumab, and you look at PD-L1 expression, you see that it really doesn't matter across here, PD-L1 expression with regard to for the response to ipilimumab. But depending on the level expression of PD-L1, you get higher response rates to the anti-PD-1. However, even if you have very high levels of PD-L1 expression, and this is now looking at tumor, and that's somewhat controversial because some people look at immune cells in the tumor, some people look at tumor cells. But if you have very high level of expression of anti-PD-1, still not all patients respond. And even if you have no expression of PD-L1, still some patients do respond. So again, some patients who have no expression can respond, and patients who have very high level of expression may not respond. And then you wonder whether you need to approach these two different groups differently in the clinic, and you have to ask the question, what is the reason for response and resistance in those different groups? So this is very nice work from Genentech and Dr. Herbst, who's a longstanding SWOG member, looking in the phase 1 trial of the atezolizumab and looking at pre and post-treatment, and they identified within the context of this trial that patients, there were basically 3 types of patients who didn't respond. Those who had no T cells to begin with and no T cells afterward. There were patients who had T cells within the stroma of the tumor. They were still there afterwards, but there was no increase in the number of T cells and no effect. And then a very interesting group in which the T cells surrounded the tumor, but did not penetrate into the tumor stroma and did not create a response. And they've come up with, Ira Melman and Dan Chen from Genentech have come up with this very nice structure to think about this, in which you can think about tumors that are, they call the immune desert tumors, for which there are no T cells, and then we have to start asking why there are no T cells in those tumors and what we could do to cause those tumors to become inflamed. There are those in which T cells are there, but they're surrounding the tumor. They just can't get into the stroma, and they're being excluded in some way. And then those tumors that are inflamed, so they have T cells, but for whatever reason, those T cells aren't enough. They're probably antigen-specific T cells in there, but they're not enough to cause the tumors to regress. The complexity comes in all the different ways one could approach this, and even within those groups, all the different ways that resistance might be mediated. So I like to show this as a graph I made several years ago, just showing all the potential stimulatory and inhibitory ligand receptor interactions that occur between an antigen-presenting cell and a T cell. And so it's not just PD-L1, PD-1, it's not just CTLA-4, those are the two that we've had in the clinic, but there's a whole host of other inhibitory ligand receptor interactions, I don't need to go through them in detail here, which could, in addition, cause suppression of T cell function within the tumor. And it's also possible, and some recent work from our Melman and Rafi Ahmed suggests that perhaps when you block PD-L1 and PD-1, that you still need a co-stimulatory signal, perhaps through CD28, in order to get those T cells to work. So the response may depend not just on blocking one or more co-inhibitory substances, co-inhibitory ligand receptor interactions, but you may also need to provide co-stimulatory signals, or they need to be there. And then you have to think about all the other factors that can inhibit T cell function within the microenvironment. So there are things like IDO and arginase, there are other cells like Tregs, myeloid-derived suppressor cells, there are type 2 macrophages, there are cytokines that suppress T cell function, and even metabolic factors, adenosine, hypoxia, low levels of glucose, and VEGF, can all impact on whether that T cell response goes on to a threshold level of anti-tumor effect. As Lillian mentioned, there are all these potential reasons, but you can do an animal model, and almost any animal model you can show that A plus B is better than A or better than B. And so this has led to an immense number of combination trials that are in the clinic now. And not just single agents, but then combinations. And this sort of falls into categories of agents that inhibit the inhibitors. And in this case, there might be things that inhibit other other things that inhibit T cell function, so other ligand receptor interactions, agents that block these suppressive myeloid-derived suppressor cells and macrophages, agents that block T regulatory cells, agents that block inhibitory cytokines, agents that reverse the effects of hypoxia. When you have hypoxia, you generate adenosine, and adenosine can bind to receptors on T cells and turn them off. And metabolic things like IDO inhibitors, when interferon gamma is produced in tumors, IDO is upregulated, and IDO can produce a substance that can be inhibitory to T cells and also can enhance formation of T regulatory cells. So you can try and block all these alone or together with anti-PD-1. And then there's the other flip side of this, which you can try and stimulate the immune response directly, which is either by vaccinating, giving substances that immunize the patient against their cancer, and that comes in all forms, including intratumoral administration or systemic vaccines, which actually try and isolate out the specific antigens that are being recognized. You can try and disrupt the barriers that block T cells from coming into the tumor, or you can do things like take T cells out, grow them, and give them back, adoptive cell therapy, or give cytokines or co-stimulatory agents. Or even more recently, you can try and take advantage of the T cells that are there but are not antigen-specific by trying to activate their, bridging them to tumor antigens and causing nonspecific activation and killing. And all of these trials and combinations are ongoing, and the estimate is there must be at least 800 or more that are out there, mostly in phase 2 trials. And again, with the problem being without good biomarkers to tell you which patients need which groups of drugs. So I'm just going to point out one combination. This is a phase 1 trial we did together with Memorial, looking at the combination of anti-CTLA-4 and anti-PD-1. This was the first combination trial of checkpoint inhibitors. We went back and looked. This is my colleagues Madhav and Kavita Dadapkar. Took cells from patients who had anti-PD-1 alone, anti-CTLA-4 alone, historic, we stored them in the spore tissue resource, or patients who had a combination, and then looked at their T cells before and after treatment and showed that if you looked at T cells and monocytes separately, gene expression in the T cells changed. It was really quite different when you gave the combination to either single agent alone, both in the T cells and in the monocytes. And when you looked in the peripheral blood and you looked at cytokine production, you saw much more cytokine production with the combination, an increase in soluble IL-2 receptor, than you did with either single agent. So this tells us that the combination is biologically active. It doesn't necessarily tell you that you'll get more anti-tumor activity. We did see activity that suggested there was more anti-tumor activity, although interestingly enough, it wasn't overwhelming, but it did suggest more activity in a sort of expanded phase 1, phase 2 trial. So that went on to a phase 3 study, and this is the recent data presented at AACR by Larkin et al., showing that, at least for progression-free survival, and again, this is not meant to be a comparison of the ipilimumab-nivolumab combination versus nivolumab alone. It's really comparing each of these individual arms to ipilimumab. But there were at least 300 patients per arm in each of these arms, and you can see that the progression-free survival here for the combination looked better than the single agent. There is some effect. It's not as dramatic as we thought we might see from the phase 1, phase 2 data. When you looked at survival, and this survival data was just presented at AACR, again, the combination arm numerically is higher than Nivo alone, but again, the differences are not dramatic. This is 2-year data, and it could be that with more time, these curves might actually separate. At least our data suggests that that's the case. But at least at this time, the 2-year survival data shows some difference for the combination. Now, this is with substantial added toxicity, so it's really important for us to figure out which patients need the ipilimumab and which patients could get by with nivolumab alone. And when you split up the two groups by PD-L1 expression, even though it's not a very great biomarker for distinguishing whether to give anti-PD-1 alone or not, it does look like it's a good biomarker for distinguishing which patients would need the combination versus the single agent. You can see that the survival curves overlap for the combination versus a single agent alone, whereas there's starting to be a nice difference here for the combination versus a single agent. In the last few minutes, I just wanted to show you that there have been a few positive signals, but with all the trials that are out there, we still don't have a huge number of positive signals. Clearly for CTLA-4 and PD-1, there have been other trials in renal cancer and non-small cell, small cell lung cancer, and some data in prostate cancer, at least preclinical data in prostate cancer suggests that the combination might be better. Anti-CTLA-4 in all the phase 2 trials that have been looked at appears to be better in combination than the single agents. However, only one randomized phase 2 trial has actually been reported with GM-CSF, which showed an improvement in survival. There's preliminary data with anti-PD-1 together with TVEC and with IDO, and phase 3 trials have been started with those. There's data with a MEK inhibitor and atezo in colorectal cancer, which looks very interesting. And some preliminary data with a few other combinations. The one that's of most interest to me has been the combinations of anti-PD-1 or anti-PD-L1 together with a VEGF receptor inhibitor. But despite all those combinations that I showed you before, so far we don't have a lot of positive signals, at least strongly positive signals in the preliminary trials. Let me skip over that. This is just preliminary data from Pembro IDO showing a higher response rate in melanoma that you would expect with Pembro alone. But look at their overall response rate was 53%. With Pembro alone, you might expect a response rate of 40%. And so this could be clearly within the confidence intervals that this was enough to go to a phase 3 trial. There have been combinations of anti-PD-1 together with anti-4-1BB, a co-stimulatory agent, and although some interesting data, these are phase 1 trials, and it's still very hard to know where to pursue these sorts of combinations in phase 2 or how to design the phase 2s in order to see incremental activity. Same thing has been true for anti-OX40 and atezolizumab. This was presented at AACR. There's only one PR in this phase 1 group that you can see here, or maybe 2 PRs. So again, not overwhelming activity and nothing compared to the really striking synergistic activity that was seen in preclinical models. What was interesting is that at least in one patient, they demonstrated, this is a patient who had had anti-PD-L1 for a while and probably didn't respond, and then added on the anti-OX40, and in this period of time had marked increase in expression of PD-L1 in the tumor, suggesting that there was a biological effect. So that kind of data might support additional study of these agents in those tumors. Since I only have a minute left, I've already mentioned the MEK PD-L1 combination, which looks very interesting preclinically, and this is the data also from presented at the ASCO meeting, showing an overall response rate of 20% for the combination in a group of tumors that you wouldn't have expected to have any response to either the MEK inhibitor alone or to anti-PD-L1. And again, that's being pursued not only in colorectal cancer, but also in multiple other diseases. There's data that chemotherapy could work together with immune therapy, and there's a lot of reasons, and again, I don't have time to go through all the rationale, but lots of reasons to believe why that might be the case. And even a phase 2 clinical trial here combining chemotherapy with anti-PD-1 versus chemotherapy alone, showing a very nice increase in progression-free survival. However, the survival curves at this point in time had not separated yet. But again, multiple studies are ongoing, and again, we don't know which patients would benefit from these types of combinations. So I'll just skip over that. I'm going to start just with this slide here. I might take one extra minute. I just want to just go through the complexity of what we have to deal with. Everybody has in their individual genetics. They've had a lifetime of environmental exposures. They have a T-cell repertoire. They then form a tumor. This tumor forms in the setting of inflammation. The tumor evolves over time. It's probably in the anti-tumor response, the immune response, is influenced by something other than the tumor itself, by host factors such as a microbiome, and that's when we see them for treatment. And if we're going to try and figure out, at a very reductionist level what they need, we would need to have all this information. We need to know everything there is to know about that T-cell response in the tumor, everything that the tumor is doing, everything else that's going on within the stroma, and everything else that's going on that's host-related, that's not tumor-related, in order to try and integrate all that information and really determine what that patient's needs. And I think that's incredibly complex, and one of the reasons that why this symposium tonight will be so important in trying to see what sorts of technologies we might have that might be able to address this level of complexity. So just in conclusion, there's a nearly infinite number of combinations in immune therapy. I don't think all the combinations need to be based on immune checkpoint inhibitors. There are patients who respond to interleukin-2 alone and don't need a checkpoint inhibitors. And even if you do use immune checkpoint inhibitors, maybe not all of them need to be based on anti-PD-1. We still don't know the critical signals that each individual patient requires in order to make that immune response lead to tumor regression, and that's why biomarkers are so important. And it may be that there are different subsets of cells within each tumor. We already know that that's the case. It may require different signals. Some of the targets that we're going after may be best to have specific delivery of these reagents to the tumor itself, and that creates another level of complexity. And it may be that scheduling may be important. It could be important to prime before you give another agent, and that's hard to address in mouse models. With combinations, just like with everything else, dose ratios may be important. That's also very hard to address in clinical studies. We know that the toxicities can be severe. We need to know what the biology is so that we can manage them better. Some patients can get very sick. And then we need to really understand, as I said before, tumor-related and host-related factors that can affect these responses. So we have an enormous challenge in dealing with all of these combinations. There's enormous promise to these therapies, but getting to the next level will be very difficult because we don't have those biomarkers. And if we're only affecting a small subset of patients in these phase 2 trials, that signal is not going to be high enough to give us the rationale to go on to phase 3 studies. So I apologize for taking a little bit more time. I'm happy to take any questions.